Imagine yourselves as bustling, self-sustaining cities where energy production and vital processes are always in motion. But have you ever wondered how the powerhouse structure in these cells, like mitochondria and chloroplasts, came to exist? The answer takes back billions of years to an incredible ancient partnership with bacteria. Let's uncover the fascinating story of the endosymbiotic theory and how it shaped the evolution of complex life forms as we know them today. The endosymbiotic theory reveals an extraordinary journey. Mitochondria and chloroplasts were once free-living bacteria that was engulfed by a larger host cell. Instead of by being digested, they form a mutually beneficial relationship with the host, eventually evolving into organelles inside eukaryotic cells. Now take a look at the image on the right. Which organelle do you think is being shown right here? Yes, it's chloroplast. What you see here is a microscopic view of chloroplast inside a beetle leaf, part of their nature remarkable energy converters. Before we dive into details, here's a roadmap for today. Explain similarity between mitochondria, chloroplasts, and bacterial cells. Describe the benefit to both an aerobic cell and an anaerobic cell that may have allowed for the formation of a mutually benefiting relationship to occur. Explain why there are two lipid bilayers around both mitochondria and chloroplasts, and how they enhance the function of metabolism. Describe the difference between endosymbiotic theory 1 and 2 and why it happened twice. Describe the societal implications resulting from this theory. Let's start with the first step, the striking similarity between mitochondria, chloroplasts, and bacterial cells. These clues are the foundation of endosymbiotic theory. Here's another close-up look at the chloroplast under the microscope. Marvel at its intricate structure. These are the factories of photosynthesis. Here is the microscopic view of a bacterial cell. And here we have mitochondria under the microscope. To fully appreciate the connection, let's break down the shape characteristic. First up, the double membrane structure. Both mitochondria and chloroplasts, like bacteria, have an outer membrane that is smooth and protected, while the inner membrane is highly specialized. In mitochondria, it's formed into crystalline to maximize energy production, and in chloroplasts, it forms thylakoids to drive photosynthesis. This structural feature is also seen in bacterial plasma membrane, which is an evolutionary clue. Now let's talk about DNA. Mitochondria and chloroplasts each have their own circular DNA, separate from the host cell nuclear DNA. This independence allows them to produce some of their own proteins and enzymes, just like bacteria do. It's like they carry a piece of their ancient bacterial identity with them. Now for ribosome. All three of them contain 70 spread-bird ribosomes, which are smaller than the 80 spread-bird ribosome in eukaryotic cell. This specific size and type of ribosome is another strong indicator of their shared ancestry. Here you can see in the picture on the right, showing the cytoplasm with rough endoplasmic reticulum and ribosome. Another similarity, reproduction. Both mitochondria and chloroplasts replicate through binary fission, the exact same process bacteria use to divide. Finally, let's talk about energy. Mitochondria are the ATP factories of our cells, converting glucose into energy through cellular respiration. Chloroplasts perform photosynthesis, capturing sunlight to create energy-rich sugars. And bacteria, many of them have metabolic pathways that mirror these processes, making them master of energy conversion too. These five shared traits are not mere coincidences. They are compelling evidence of an ancient union between early eukaryotic cells and bacteria, which transformed life on Earth. Now that we understand mitochondria and chloroplasts evolved from bacteria, let's dive into how this incredible symbiotic relationship may have formed. To start, let's distinguish between aerobic and anaerobic cells. Anaerobic cells are organisms that cannot use oxygen to generate energy. Instead, they rely on processes like fermentation, which are far less efficient at producing ATP, the energy currency of the cell. On the other hand, Aerobic cells can use oxygen for energy production through a process called oxidative phosphorylation, which generates significantly more ATP than fermentation. Imagine an ancient aerobic cell living in a world where oxygen levels were rising. For anaerobic organisms, oxygen was toxic because it led to the formation of harmful reactive oxygen species. However, aerobic bacteria had evolved mechanisms to metabolize oxygen safely and efficiently, giving them a survival advantage. When the anaerobic cell engulfed one of these aerobic bacteria, something remarkable happened. The anaerobic host cell gained a powerful advantage, the ability to produce ATP much more efficiently through the bacterium oxidative phosphorylation process. This extra energy allowed the host to survive in environments where oxygen was becoming more abundant. In return, the anaerobic bacterium benefited too. It found a stable home within the host cell, with access to plenty of nutrients and protection from external environmental threats. Together, they form a mutually beneficial partnership, a relationship so successful that it eventually gave rise to mitochondria in eukaryotic cells. This ancient union didn't just help the cell survive, it paved the way for the evolution of more complex, energy-efficient life forms. This is how a simple interaction transformed the life on Earth forever. The double membrane structure of mitochondria and chloroplasts is the direct result of their evolutionary origin through endosymbiosis. Here's how it happens. When the ancient host cell engulfed a bacterium, the outer membrane of the bacterium became the inner membrane of the organelle, while the host cell or membrane formed the outer layer. This double membrane reflects their shared history. But why is the double membrane important? It enhances the organelle function in metabolism. In mitochondria, the inner membrane is folded into cristae, increasing the surface area for electron transport chain to generate ATP. In chloroplasts, the inner membrane surrounds the stroma and thylakoid system, allowing light-dependent and light-independent reactions to occur efficiently in several compartments. This compartmentalization and increased surface area are crucial for energy production, making these organelles highly specialized and efficient. Now let's take a closer look at the two key events that led to the formation of mitochondria and chloroplasts. The first event, endosymbiotic theory, occurred when an ancestral eukaryotic cell engulfed an aerobic bacterium, likely an alpha-proteobacterium. This bacterium eventually became the mitochondrion. With this new partnership, the host cell could efficiently produce ATP using oxygen through aerobic respiration, which was essential as oxygen became more abundant in the environment. The second event, endosymbiotic theory 2, it happened later when a eukaryotic cell that already had mitochondria engulfed a photosynthetic bacterium known as cyanobacterium. This bacterium evolved into chloroplasts, allowing the cell to perform photosynthesis, which enables it to harness energy from sunlight and a crucial development for plant cell and some protists. Now you might wonder, why this process happened twice? Well, the answer lies in the distinct function of mitochondria and chloroplasts. Mitochondria are involved in energy production in all eukaryotic cells, whereas chloroplasts are responsible for photosynthesis, a process specific to plants and some protists. These events occur sequentially because the first endosymbiotic event provided the early eukaryote with a more efficient way to generate energy in an oxygen-rich environment. The second event gave rise to chloroplasts, which enables cells to use sunlight to generate energy. Genetic evidence supports this idea by showing that mitochondria and chloroplasts have different evolutionary origins. Mitochondria are related to alpha-proteobacteria, while chloroplasts are linked to cyanobacteria. This confirmed that the endosymbiotic process occurred twice, each with a distinct purpose. Building on this idea, let's explore the societal implication of the endosymbiotic theory. How does this theory challenge our understanding of evolution? And what impact does it have on the fields like medicine, technology, and environmental science? The endosymbiotic theory provides a profound shift in our understanding of the origin of complex life. It suggests that mitochondria and chloroplasts, essential organelles in eukaryotic cells, were once living prokaryotes that entered into symbiotic relationship with early eukaryotic cells. This challenges the traditional view of evolutionary competition and emphasizes the cooperation as a central force in biological evolution. This perspective has far-reaching implications, especially in the fields of medicine and biotechnology. For instance, a deeper understanding of mitochondrial evolutionary origin aids in the research of mitochondrial diseases and could guide the development of targeted therapies. Furthermore, this theory has spurred advances in biotechnology, including the manipulation of organelles for applications like biofuel production and enhanced agricultural practices. By recognizing the importance of these ancient symbiotic events, we can better address modern health challenges and ecological needs. Beyond its impact on medicine and technology, the endosymbiotic theory also offers important lessons for environmental and philosophical thought. It underscores the role of microbes in sustaining life ecosystem, which may encourage greater efforts toward biodiversity conservation and environmental protection. This theory also has profound philosophical implications, suggesting that the interdependence, rather than competition, is a fundamental principle of life. This insight could inspire new approaches to global challenges like climate change, where cooperation between nations, ecosystems, and species is essential for addressing shared problems. However, with the growing potential to manipulate organelles or even create synthetic organisms, there are significant ethical considerations. As humanity gains the ability to enter fundamental aspects of life, questions arise about the boundaries of scientific intervention and ethical responsibility involved. The endosymbiotic theory therefore not only reshapes our biological understanding, but also prompts critical discussion about science, ethics, and our role in shaping the control of life on Earth. Thank you for your attention.